Thank you for joining us. I am Juniper from Hawaii.
I'm Andrea. I'm from Kenosha, Wisconsin.
And I'm Kelley from Connecticut.
This past month, our research revolved around studying a bottom-up control ecosystem during the spring bloom. As you've heard before, phytoplankton are among the most important organisms on Earth. They are known as drifters because the currents of the ocean force them around and they have no ability to move. They are the base of the food web. Everything else, even organisms on land, depend on them. There are two types of plankton, phytoplankton and zooplankton. Phytoplankton are plant-like plankton that photosynthesize to make their own energy from the sun. They are primary producers. Through this biological process, they convert carbon dioxide from the atmosphere to 50% of the oxygen we breathe. Zooplankton are animal-like plankton and primary consumers. It is important to study the relationship between phytoplankton and zooplankton because they are the first link in the food web and can tell us whether the ecosystem is in top-down or bottom-up control.
So all ecosystems are controlled by either the top-down or the bottom-up, meaning that either the consumers or the producers are controlling the population of the food web. Bottom-up control is the most common. This is when the producers are controlling the population of all the consumers above it on the food chain. For example, zooplankton eat phytoplankton. If there is a high population of phytoplankton, then it will cause the population of zooplankton to increase as well, because there are enough producers to allow for more consumers. This will continue up the food chain until it reaches the top predators, making their population grow as well. The opposite of this is top-down control. This is when the predator population controls the population of all the prey below it. For example, if there's a large population of zooplankton, then the population of phytoplankton will decrease because the phytoplankton cannot reproduce as fast as the zooplankton are consuming. Many factors contribute to whether or not an ecosystem is under top-down or bottom-up control. One of them has to do with the winter-spring bloom.
The North Atlantic spring bloom occurs as the seasons change from winter to spring. It is a strong increase in phytoplankton abundance and diversity as a result of environmental conditions like a light availability, nutrients, temperature, stratification of the water column. This increase is so great that you can see it from space and is an important part of ocean production. The image above shows chlorophyll amounts in the North Atlantic Ocean. Chlorophyll is a proxy of photosynthesis, so this is showing the abundance of phytoplankton during the bloom. The closer to red the color, the higher the amount.
There are two hypotheses that explain the bloom. The first is shown in this diagram called Sverdrup's critical death hypothesis. In the winter, we see a mixing of nutrients and plankton, and it really replenishes the nutrients in the surface layer where the plankton thrive. In the spring, when we have more sunlight and runoff from the snow, we see some layering in the water column and a large increase in plankton because of the nutrient-rich water and access to sunlight. The second hypothesis is the dilution recoupling hypothesis. In the winter, the plankton are spread out over the entire water column, so there are less predator-prey interactions. But in the spring, they're living in that small little layer, and so there are way more predator-prey interactions, which might cause more plankton in the spring. Early in the spring, we probably see more phytoplankton and less zooplankton, but later in the spring, when the zooplankton have had some time to catch up, we'd see a little bit less phytoplankton and more zooplankton, and that would be an example of top-down control. And this graph clearly shows stratification of the water column with the cold water at depth and the warm water at the surface. You can see the colors there. Okay, these profiles show temperature. The line and the color both show this. And as you can see here, the difference between the winter ocean and the spring ocean is really temperature, and then salinity too. And if you remember what this looks like, we'll see something just like it again.
So So after studying how important plankton are to the ecosystem and how we can predict the trends of the food web just by looking at the first link between phytoplankton and zooplankton, we asked ourselves the following 3 questions. Do we see variation in abundance with different physical and environmental factors? Do we see evidence for top-down or bottom-up control? And how diverse are phytoplankton and zooplankton during the winter-spring bloom period?
We hypothesized that there would be more phytoplankton than zooplankton across all locations. This would be an example of bottom-up control. We also expected higher diversity of the phytoplankton than the zooplankton.
So we went to 3 different locations to collect our data. First, we went up north to the Maine Maritime Academy in Castine, Maine. While there, we sampled at 2 locations, a stratified area down the Penobscot Bay and a well-mixed area up the Penobscot Bay. Then we took another well-mixed sample at the South Freeport Town Dock, as shown in the maps.
We used a CTV to measure salinity, temperature, and depth, and we used that to collect some of our samples. And we used an 80 micron net tow, which really collects more zooplankton, and the 20 micron concentrator, which really focuses on the small phytoplankton.
Once back at Coastal Studies for Girls Laboratory, we used a counting slides and a microscope to count and identify the amount and types of plankton in each of our 6 samples. Once we had done this, we used analytical methods to calculate the Simpson's diversity and cells per liter abundance for each of the samples.
We had the opportunity to sample at a stratified location and a well-mixed location. As you can see, the well-mixed location, as the CTD goes up in depth, we see no change to the temperature, which is in blue, or the salinity, which is in red. In the stratified sample, as the CTD goes up in depth, we see major changes to temperature and salinity.
These graphs above show the abundance of cells per liter based on each of the samples. And the dark blue and dark red show the 80 micron samples, and the light blue and light red show the 20 micron samples. The blues represent phytoplankton, and the reds represent zooplankton. On the y-axis is the logarithmic order, the logarithmic scale, and on the x-axis is the location. In any 1 liter of seawater, there can be anywhere from 10,000 to 10 million phytoplankton and about 100 to 1,000 zooplankton. This is about the amount in 1 water bottle or an algae. This is an incredible amount, as you probably can see. We can see that there is more zooplankton in the 80 micron samples, shown in the dark red. There is hardly any zooplankton in the 20 micron up bay and down bay Penobscot samples, shown by the lack of the red bars under, the Penobscot Bay locations. And in both graphs, we can see that there is the most plankton, zooplankton, in the town dock samples. Also, the up bay and down bay samples are very similar, while the town dock sample is very different. This may be due to the difference of the locations of those samples and those locations' conditions. Looking back to the question, are there more phytoplankton than zooplankton? We can see that in all the samples, this is true. There are more phytoplankton than zooplankton, represented by the blue.
So after looking at the abundance of phytoplankton and zooplankton in general, we decided to zoom in and look at the different species. These graphs show the type of plankton found in both the 80 and 20 micron samples. The x-axis shows the different locations, and the y-axis shows the cells per liter abundance in logarithmic scale. The different colored bars are representative of the different species. As you can see, there's much more diversity found among the phytoplankton than the zooplankton. We only found a total of 4 different zooplankton species: copepods, nauplii, brittle star larvae, and amphipods. Whereas we found 7 different species of phytoplankton: Thalassiocyra, pennate diatoms, Cattosaurus, diatoms, centric diatoms, Rhizosolenia, and Hemiolus. Here are a few of the different species we found. higher diversity of phytoplankton than zooplankton is important because it tells us what kind of diversity we can expect during the winter-spring bloom. What we found really interesting was that we see a small amount of top-down control between the individual phytoplankton and zooplankton species. For example, the 20 micron up bay sample has the highest diversity of phytoplankton and no zooplankton. But then the down bay and town dock samples have a lower diversity of phytoplankton and more zooplankton. This tells us that when there's more phytoplankton, there is less zooplankton and vice versa. This is a form of top-down control on a small scale, but then we wanted to look at the big picture. If you can recall, one of our research questions was looking at whether or not the whole ecosystem was in top-down or bottom-up control. The last diversity graph shows us that there's a top-down control between species, but our data at these locations this data shows us that all locations are in overall bottom-up control. What you are looking at is the ratios between phytoplankton to zooplankton in all of our samples. The phytoplankton are shown in blue, and the zooplankton are shown in red. As you can see, the phytoplankton are much more abundant than the zooplankton in all of our locations, especially the up and down bay samples we took at Maine Maritime. We believe that this is because we are sampling at the end of the winter-spring bloom, and the zooplankton have not yet been able to receive the effects. However, from the looks of our data, the zooplankton had started to increase in abundance at the town dock. This may be due to the fact that we took the town dock sample about 4 days after we took the Maine Maritime samples, which could have given the zooplankton more time to increase. This also could have been due to the fact that Freeport is located further south than Maine Maritime, which means that the bloom could have occurred earlier in Freeport. But whatever the case may be, it shows that the ecosystem is indefinitely in bottom-up control because the high population of phytoplankton is causing the population of zooplankton to increase as well.
So, does stratification affect abundance? No, we saw no trends relating stratification to abundance, but we did see a lot of phytoplankton across all locations. Does stratification affect diversity? No, we saw no trends relating diversity to stratification. And what types of control did we see? We observed a lot of overall bottom-up control, and we saw top-down control between the specific species.
So what does this mean for our future of the oceans and ourselves? Because there's more bottom-up control overall, the health and survival of the phytoplankton is important because they support the rest of the food web. If there are lower amounts of them, then there will be less support for the populations of other organisms in the food web, including us. Even though we saw no correlations between the stratified and well-mixed samples, stratification of the ocean will become more prominent as climate change continues. As there will be more stratification, the plankton will be stuck in this layer with only the nutrients that are already there because of the lack of mixing between layers. They will first increase in abundance and diversity, but then use up all the nutrients in the layer and the population will decrease. The North Atlantic spring bloom occurs every year, so we can expect that there will be increases in populations of all the ocean ecosystem organisms during this time. Lastly, we would like to thank Carrie Whitaker for her continuous support and guidance with data collection and analysis.
Thanks to Laura for waking up early with us to collect our data and for helping us through our data analysis.
Thank you to our fellow scientists and classmates for helping us collect our data.
We would like to thank Maine Maritime Academy for allowing us to use the research vessels and supplies to collect samples.
Thanks to GMRI for having us tonight.
And thank you all for coming.